Okay, so let's talk about meiosis and sexual reproduction. This is the third part for this lecture set. The first part we were just talking about overview of meiosis and which types of cells go through it. The second part we were looking at meiosis 1, the first part of meiosis, which consists of prophase 1, metaphase 1, anaphase 1, telophase 1. So we left off with this diagram, this picture here, kind of looking at the overview. And remember when we left off, we talked about this thing called interkinesis. That happens at the end of telophase 1. Interkinesis is when the cell goes through meiosis 1, and now it says, okay, I'm done. Time to take a break. I'm going to take a little bit of a resting period here and pause. After it pauses, it then starts up and goes into meiosis 2. Now, meiosis 2 begins with prophase 2. So, both of these cells will simultaneously be going through prophase 2. So again, look for key events. What's going on with the chromosomes? So the chromatin condenses back into the chromosomes. The nuclear envelopes dissolve. The centrioles appear. Everything's getting ready and gearing up for the next stages. But a big thing to keep in mind here, there is no crossing over during prophase 2. The cells have 23 chromosomes in each of them. They do not pair up and they do not cross over here. They only cross over in prophase 1. Okay, so there's no crossing over happening here during prophase 2. From prophase 2, each cell, both of these, will at the same time go into metaphase 2. Metaphase 2, they're going to line up, but each cell contains 23 chromosomes. Okay, so 23 chromosomes in each cell. So cell right here, 23. This bottom cell, 23. 23 chromosomes in each of these cells that will line up during metaphase 2. The cell then moves to anaphase 2 and it pulls apart those chromosomes. So now each cell will have 23 chromatids moving to the right. And 23 chromatids to the left of each cell. Okay, so we're going to have 23 going this way, 23 moving down towards that bottom spindle fiber, and then the same thing in the other cell. So 23 chromosomes have been pulled apart in each cell. Now we have 23 chromatids going to the right, 23 to the left, or 46 total chromatids that should, here's the big key, should be divided evenly, going to the right and to the left in each of those cells. If they're not divided evenly, that's when we get into some of these genetic disorders. And we'll talk about that in the next lecture and as we go further into this information. Okay, so anaphase 2. Ana means apart. The cells are going to start to pinch and divide themselves in half. Telophase 2, nuclear envelopes reform. Cell continues to squeeze and pinch in half, dividing it. And now we are at the end of telophase 2. We should be looking at 4 new cells that are genetically unique. Okay, each of these cells was going to have different pieces of genetic information. Each cell will contain 23 chromatids, But because of crossing over all the way back in prophase 1, those chromatids are all mixed up. What piece of information is found on each chromatid is going to vary between each of these cells. So this is why you don't know which of these cells is going to become the egg or which one will become the sperm that actually gets to the egg. That's why there is the statistically, it's pretty much impossible to produce identical children from two different reproductive events. Okay, so telophase 2, at the end here, we have 4 new cells that are genetically unique. And let me underline that just to reinforce genetically unique cells. Okay, so here's our overview of meiosis 2. So prophase 2, you have 2 cells, chromosomes condense, they get ready, everything's getting organized. Metaphase 2, 23 chromosomes line up across the center of that cell. And then we go into anaphase 2. Let's pull apart the chromosomes, and now we're calling them chromatids. Half of them go to one side, half go to the other side. That happens simultaneously in both of the cells. And then ultimately at the end, 4 cells that are genetically unique. Okay? Again, the key being genetically unique. So, as we were saying, we have 4 cells at the end here. Which one's the egg? You got a 25% chance. Which one is the sperm that fertilizes the egg? 25% chance from that grouping of cells. So these are all germ cells that go through this pathway, that go through this process to become eventually an egg or sperm cells. So here's our overview, looking at meiosis and comparing it to mitosis. Meiosis 1, there's a lot of similarities between meiosis 1 and mitosis with what's going on. Chromosomes condensing, they're getting lined up, they're getting organized, they're being pulled apart, etc. So the order of events is the same. Prophase 1, metaphase 1, anaphase 1, telophase 1. There's 2 daughter cells at the end. Big difference though, our daughter cells at the end of meiosis contain half the genetic material, whereas in mitosis, at the end, they have half the genetic material, but then they're going to go and replicate it. We don't do that in meiosis. So I said use this comparison to just get familiar with, okay, here's some of the similarities, but let me put some little stars in here for you guys. Make sure you're comfortable with what are the differences. Know the differences. What are the key differences between mitosis versus meiosis? What's going to separate those out if we're looking at them? So definitely something I want you guys to look up. Key differences. Between mitosis and meiosis. Alright, that's going to definitely be an important factor, important piece of information for you guys to explore. Okay? Alright, so here's just meiosis 2, what's going on, just re-emphasizing it. Prophase 2, metaphase 2, anaphase 2, telophase 2. As they go through this, we wind up with 4 daughter cells that each contain 23 chromatids. We're going to call those daughter cells haploid. So diploid refers to full amount of genetic material. Haploid refers to half. So let me just re-emphasize it. Haploid cells at the end of meiosis 2. And haploid cells are represented as little n at the end of meiosis 2. So mom's doing it, she's making haploid cells. Dad's doing it, making haploid cells. Those 2 cells come together, they form the zygote. The zygote now is diploid. It has 2n, all the genetic information that it needs, hopefully, to function correctly. Okay, so again, just looking back, overview here, not going to spend the same amount of time, but just another comparison, prophase 1 to mitosis, the prophase aspect of mitosis, metaphase 1 to mitosis, metaphase, anaphase 1, telophase 1, those comparisons to what's going on in mitosis. This one here is just another comparison of the two, looking at what's going on between meiosis 1 and mitosis there. Alright, so just again trying to use the visual diagrams to help make it a little easier for us. So, alright, now when this goes on in guys, this is called spermatogenesis. This is the production of sperm in males. So one sperm, or the cell, the primary spermatocyte, this is the germ cell right here, starts out as 2N, haploid cell, or diploid, I'm sorry, diploid cell, 2N. When it goes through meiosis 1, it rips in half, and now it becomes haploid. It's got half the genetic material. It's called a secondary spermatocyte. Those go through meiosis 2, and they divide even further down with their genetic information into what we call spermatids. Now, each of these spermatids should contain 23 chromatids. Should have 23 in each of these little spermatids. Those are sitting in the testes. They'll sit there for so many days, and then they move into maturation, become mature sperm. They get their tail, their little a chromosomal cap, little enzyme that helps them actually get into the egg. And these guys are going to be ready to go. Each one of these is genetically unique. Each sperm cell has a slightly different genetic code of information. And again, keep in mind the term haploid. That's what we want to use to describe the sperm cells, is that they are haploid cells. Okay? So when we're looking at the haploid sperm cells, spermatogenesis is the process. Oogenesis is the process of forming an egg in moms. Okay, so same process, same idea. Start out with an oocyte, which is diploid. That oocyte divides in half. But the big difference here is the division is not equal when we talk about the amount of cytoplasm and Golgi bodies and mitochondria and cellular components. It's an uneven division. So we have one large cell called the secondary oocyte and a smaller cell called the polar body. Both of these contain 23 chromosomes. They contain half that information. They are not going to be ready to do anything for quite a while. They'll sit there in the woman's body until possibly fertilization occurs. Then those cells can divide again. They are, all right, let's go through meiosis 2. Let's complete that process and become the egg cell. So now it divides again, and it produces an uneven divisioning, and that uneven divisioning produces a second polar body, and then the big, large cell here that has all of the, or the majority of the cellular components, the genetic material, etc. Sometimes that first polar body will divide and turn into another polar body. Depends on the cell, depends on the division. But now we have the haploid egg, the haploid sperm, that fuses in fertilization, forming the zygote, which goes back to being diploid. All right, so I'll diagram this out and have you guys diagram it. It usually works a lot better to visualize oogenesis and spermatogenesis in a drawing. But this is going to start at puberty. So whenever that young lady started puberty, whatever age, 12, 13, 14, sometimes we hear about 8 or 9-year-old girls starting puberty, same thing for guys. That's when this pathway starts and it finishes. Your body runs out of germ cells when you go into menopause. Okay, so it does eventually end, and we do not produce viable sperm or viable eggs after our body has shut down. yet the cell cycle keeps going until the day we die.